Hey guys, Dr. B. This is just a little exam review study guide to kind of focus your, learning for your exam on Wednesday. So first things first, obviously there's a lot of things in red. Those things in red are worth more points than the things in black. The characteristics of viruses, obviously, like what makes them different from living cells. There's a lot. So just be aware of how they're different. And their basic structure, how they're organized. Obviously, the next one, steps in viral replication, is pretty important stuff. You definitely want to know the way that animal viruses replicate different than how bacteriophages replicate. Do not confuse those terms because they replicate in different ways. And so be able to obviously compare and contrast those. The next three bullets are basically just, or next two I should say, are pretty straightforward. I mean, you just need to know what those are. So not to say that, hey, straight up memorizing, but those are, you need to know what a mesophile is and thermophile, and examples of them. Moving down the list of fusion osmosis, you're going to be put into scenarios where you have to tell me whether a cell would shrink or swell or stay the same given a certain environment. And that kind of tests your conceptual understanding of what osmosis is, and diffusion as well. So again, moving down, these are again more just know what they are, and examples as well. Function control enzymes, pretty fundamental. That's not a ton of points. That's probably the least amount of points of anything in red. But you know what controls enzymes and whatnot. Cell respiration, obviously a large topic. So the biggest way to do this is to just kind of obviously watch my lectures on them. But what goes in and out of every stage is really important. Like numbers of things like, you know, CO2s and ATPs produced and things like that. You do not need to, for the most part, memorize numbers of like NADs or NADHs that are, you know, produced at any given step. I never found that to be very critical to the understanding of cellular respiration. At least the numbers of them, obviously know what their function is. So that's pretty straightforward. You need to be able to recognize that, and also don't forget fermentation as well. Obviously, when there's not enough oxygen around, and you know some cells can just repeat glycolysis and pyruvate production over and over. You know, and some cells like yeast, for example, can produce ethanol and carbon dioxide, and animal cells can produce lactic acid and carbon dioxide. So have a kind of a understanding of that. So let's get to a couple exam questions. This is the typical question you'd have. Again, it wouldn't necessarily be number 41, but you know, just an awareness of kind of what goes in and what goes out of stages of glycolysis. That's pretty straightforward. And when you look at this question, you're like, I don't remember talking specifically about the formation of peptide bonds between amino acids. That doesn't mean that this is not a fair question. So understanding the idea of what a formation of bonds would be in terms of those examples is the key to that question. Last but not least, a good question that kind of has, you know, testing the your understanding of what a barophile would be, obviously barometer. So you can go ahead and figure that one out. But a lot of these are like examples of the understanding. So hopefully that helps. In terms of the open-ended stuff, there'll be a chart with cell respiration where you fill in some parts of it. I'm not going to show it to you, but again, straightforward. So that just should give you guys a little bit of an idea about what you're going to see. And exam is on Wednesday, so I'll see you then. Email with any questions.